Computational Biology by Mr. Shounak Sanyal. We are delighted to have you here. I would also like to extend a special welcome to our chief guests, Assistant Professors Dr. Deepthi and Dr. Lini. Let's also take a moment to acknowledge Devadarshan, our moderator, and Tirtha, the creator of this wonderful group, for their efforts in bringing us all together. And I'm Akshaya, a postgraduate student of zoology from Sacred Heart College at Ernakulam. I feel extremely privileged to be a part of the session and to host this session today. Our webinar in computational biology will delve into the fascinating world of this field. We know computational biology is where the intersection of biology, computer science, and data analysis happens. In today's world, where biological data is peaking or growing at an exponential rate, computational biology acts as a bridge to unravel the mysteries and to derive the insightful insights. Whether you are new to this domain or have already been experienced in this, I'm sure today's session will resonate with you in some way. Now, the most awaiting session, let me introduce our distinguished and esteemed speaker, Mr. Shounak Sanyal. He is currently pursuing his MTech in Bioengineering at the Indian Institute of Science, Bangalore, and has achieved the remarkable feat of securing the All India rank of 11 in GATE Biotechnology 2023. A graduate of NIT Raipur and a proud Kolkatan, Shaunak also completed his high school education at Mansoor Habibullah Memorial ICSE School, Kolkata. An avid reader and a brilliant learner, Mr. Shounak Sanyal's passion for knowledge and his ability to share it effectively make him an exemplary guide. Without further ado, let us welcome Mr. Shaunak Sanyal to take the spotlight.
I hope I'm both visible and audible to everyone. Um,
Yeah, you're audible.
okay. Thank you, Akshay, for that introduction. It's a bit flattering. So, hello everyone. I'm Shaunak. I am an MTech student, and I'd like to talk a bit about my experience in this new field of computational biology. When I'm saying it's a new field, I mean to say that compared to the other more mainstream, more conventional fields of biology that we are aware of, computation, especially the application of mathematics and physics, even some data sciences, these things are emerging. And if you look at in the past couple of decades, you'd see that a lot of progress has been made in this field, and it's what you call a hot topic in academia these days. So I'd like to share my screen now. It's not a PPT. Is my screen visible? Can anyone confirm if my screen is visible?
Yes, it is. It is visible.
Yeah.
Okay. I'm not keeping it in slideshow because I have to toggle a bit around. So, like the title suggests, welcome to my presentation. I'm gonna walk you all through what I've learned and my experience in mathematical biology. Now, I would assume that most of you are students here, and like just like you, I'm a student myself. I'm not an expert in this field, but I feel that I've had a taste of this subject, and it has occurred to me that it would have been rather more convenient had I have more knowledge in this field even a couple of years back. I feel that mathematical biology is a very niche topic, which about which not a lot has been discussed in an undergraduate level. So this is just an attempt to walk you guys through this area, just to make you aware that such a field even exists. And this is not at all going to be a very detailed session. I'm not going to go into the math. In fact, I'm not at all going to go into the math. I'm just going to show you a few instances where you can find that mathematical theories, which we normally associate with something very abstract, or theories from physics, which we normally associate with things like our galaxies or electronics or quantum mechanics, you can sort of find them existing in life sciences as well. And as someone who has been from a biotech background, I find it to be very fascinating that concepts which I never thought would have a sort of a coexistence can sort of merge together and form this really new, exciting interdisciplinary field. So, without further ado, I will start off. So, biology, as I have observed, and I think most of you would agree with this, is that it's been sort of categorized into two domains. One is a more fundamental, where we are more interested in studying the biology of things. So maybe we are interested in understanding how a cell works. We're interested in understanding the biochemical pathways of the different biochemical procedures. We might have an inclination to study microbes. And in most of the times, at least, the motivation to study these things was because it was there. However, in the 21st century, applicative biology is more pertinent and it has a lot of opportunities to offer. So you have fields like biotechnology, bioprocess engineering, and systems biology, therapeutics, drug delivery, all of which use the knowledge of biology, and they try to sort of put it into a context where you can make something out of it. You can make a product out of it. You can design a therapy out of it, or so has been the motivation. Now, math or mathematical biology sort of falls into the interface of both of these areas. And I have a few slides dedicated just for that. But when I was first introduced to this subject, I sort of had two questions: that why do I care about the math? I mean, what am I going to get out of that? And I have got a really nice video about fireflies, and I think that would serve as a really that would give us a very good motivation as to why should we care about the math. Now, I simply before I start the video, I'd like to sort of give a small premise. So fireflies, they emit light, and they mostly do it to communicate or to mate with each other. Now, there are mathematical equations that govern how the fireflies sync or synchronize with each other in a community. Now, the mathematics is so accurate is that a group of scientists from Thailand, they tried to use LED lights, and they tried to see that. I'm sorry. Does anyone have anything to say?
That's okay. I believe there was a
Uh,
disturbance.
okay. So as I was saying that an attempt was made to use LED lights uh, and try to synchronize the fireflies in a mangrove forest using the constant shimmering, the glowing of the LED lights. And as you would see in the video, that they were able to successfully do it, and that sort of proves that the underlying mathematical equation actually works in explaining a situation like that. So I'll start the video for you. I think it would be much better if you see it by yourself. I hope you all can see it.
Shanak, does the video have an audio?
Yeah, I'm sorry, I didn't see it. Yeah. Right. So I hope you guys could see how especially in the later part of the video, all the lights were sort of glowing and dimming down in a very oscillatory manner, in a very periodic manner. And it was very hard to sort of distinguish between the LED lights and the fireflies. And this sort of shows that this is how this is how effective mathematical modeling could be. And as you could observe, the equation here is the Kumamoto model, is essentially the governing equation, which we usually use to sort of explain how the fireflies would sink. Now, I won't go into the details of the model. The reason I've kept this equation is for you to just appreciate the fact that there is something mathematical at work here. And another interesting thing is that the Kumamoto model, which is essentially, like I showed you, an application is used to explain fireflies, is not just limited to that. So in our heart, there are certain regions in our heart known as the sinoatrial portion of our heart, that is made up of a lot of cells, and those cells essentially govern the beating of our heart. And I'm pretty sure you're aware that our heartbeats also happen in very periodic manner. It has got a certain pattern associated with it. You can use the Kumamoto model to explain why the cells in the sinoatrial region of our heart behave exactly in a very synchronized fashion to produce the signals that in turn go to the ventricles and result in the heartbeat. So, uh, a concept or a phenomenon that is observed in fireflies can also be translated into explaining the pacemakers of our heart. But it doesn't end there. In epilepsy, which is a mental condition, it's a mental health condition, there are certain cells, certain nerve cells in our brain that also tend to occur in a sort of a synchrony. That again, which can be explained by the Kumamoto model. Although that sort of synchrony is detrimental to the human beings, but the underlying fact is that synchronization, about which there is already there is a huge amount of mathematical literature available, can be so conveniently utilized and manifested in these seemingly distinct fields. And as biologists or people from biology backgrounds, we are not supposed to know it because we are not mathematicians. But and this is exactly where the role of mathematics or mathematicians come to play, is that they sort of act as a glue that sort of brings together all of these underlying concepts and shows this one overarching phenomenon, using which you can sort of appreciate that one theory or one idea is getting sort of utilized in so many different fields, in so many different areas. And by the way, if I'm going too fast or if you guys have anything to ask, please feel free to interrupt and ask it. So I'll move on to my subsequent slides. Yeah. So, like you observed in the video, that we started off with fireflies, something which is got to do more with ecology. And as I've observed in my limited uh, field of exposure that I've done so far, is that a lot of mathematics that has been sort of diffusing into more cellular or biological systems are mostly inspired from pre-existing ecological models that have been initially used to describe, let's say, the life cycle of organisms or species in an ecosystem, or the distribution of species or organisms in an ecosystem. And and this is something which I really find very very intriguing, is that ecology is almost acting as a muse for applied mathematics in a biological context. And a very notable example would be the Lotka-Volterra model, about which I'm going to talk about in the next slide. So, okay, so there's a lot of information there. Please don't get bogged down by it. The Lotka-Volterra model is a system of differential equations that basically help us to understand the dynamics of certain species in a population, especially those species or organisms that have a predatory prey nature with each other. So, In case you are not aware of differential equations, they are essentially mathematical equations that are used to govern the rates of certain quantities. And that could be anything, including but not limited to the number of predators and prey in, let's say, a forest. Right? So X here is the predator and Y here is the prey. So what's happening is that the predator is eating the prey, the prey is being eaten up by the predator, the prey is eating some resource, and both the predator and prey, they are dying. I hope you could follow me. I'll just repeat once again. The predator is eating the prey is being eaten up by the predator, the prey is eating some resource. So if you can imagine a tiger and a deer, so the deer is eating some grass, and other than that, both the tiger and the deer, they are also dying by some natural causes other than the tiger eating the predator. So I mean, sorry, the tiger eating the prey. So the deer has two ways of dying. One, it can be eaten up by the tiger, or it can die naturally. And the tiger has only one way of dying, it dies naturally. So that's been a lot of words, but if you want to sort of bring them down to a single or a set of mathematical equations, you'd use something known as a Lotka-Volterra model. And if you could get your focus on the image here, I'm not sure if my cursor is visible, but you could see that there is a sort of a periodic curve with blue and oranges. Basically, if you solve those two equations above, the result would be something as you would observe in the left, in the bottom left. And the orange is the predator and the blue is the prey. And as you can see is that there is a constant oscillation between the two. So when there is a lot of predator, consequently the prey population will fall, and if the prey population falls, the predator would also fall. So there is a very distinct relation between the predator and the prey. And if you use something known as a phase diagram, which again it's a bit more mathematical, but a phase diagram is essentially used to check how a set of differential equations behave. You'd find circular features as is observed in the bottom right. Now, all of these diagrams and static images might be hard for most of you to appreciate. So I have another video for you where a simulation has been generated using the same principle, and where there is one resource. I mean, sorry, there's one predator and one prey, and we'll observe how the the numbers, the levels, the concentrations, if I could say it like that, they sort of oscillate and move up and down in a very systematic fashion. So I'll play the video for you. So here the red is the predator, the blue is the prey, most probably. And you can see that in different locations throughout the grid, you're observing series of rises and falls. And that's exactly how you would expect a predatory prey model to work. I'll just let it run for a couple of more seconds. Right. So, like I mentioned, that this is an ecological model. This is a model that you can use to explain species and organisms. But is it pertinent in something else? As it happens, it is. And well, I don't have a lot of slides or images for that. So, unfortunately, you guys have to visualize that. So let us suppose I have a protein. Let us suppose I have actin. I'm pretty sure a lot of you have heard of actin. And as it happens, that actin is often, how do I say it? F actin, which is a part of actin, often undergoes a change in form. So it can become branched actin, or it can become protosomes, or it has to traverse throughout the cell. So this chunk of F actin that's there is been utilized in these different many forms. And if you let your imagination run a bit, you can sort of draw parallels where you can think of F actin to be a resource, and the protosomes and the branched actin to be two species that are consuming the resource. And perhaps there is also a conversion between the F actin, between the branched actin and the proteasomes and so on. So that would add a bit of competition there. I'm not sure if I'm able to sort of motivate you to develop the motivation behind this, but the point being is that you can use a Lotka-Volterra model in a system like that as well, where you can observe how one F actin is getting converted into multiple different forms, and is there a competition between those two forms with the F actin? If there is a direct competition between the two forms, is one product sort of diminishing the other, or is one product activating the other? So these are very very interesting areas where you can sort of go through, with your motivation being the Lotka-Volterra model and related ecological models. So that's how the ecology. I mean, a lot of studies of ecology are being very pertinent in more mainstream biological fields. Right. So I've been talking a lot about models. Now, before I go, so I have a couple of more sort of slides where I'll have a couple of more examples. But before that, I want to stress onto the fact that mathematical biology, at least, is all about designing models, and you can sort of draw parallels with it from concepts of physics. So what happens in physics is that we create a hypothesis. A hypothesis is basically it's a framework, again a model. You use mathematical equations to describe that, and and if you're lucky, you'll probably have an experimentalist, or if you're an astrophysicist, you'll have an astronomer or a cosmologist to carry out experiments, uh, based on which you can sort of validate whether or not your theory is uh, whether or not your theory actually works. A very good example was the firefly example, where I showed you an equation, and then a very ingenious experiment was designed to test the validity of the model. Another example would be the theory of relativity, which Einstein had done entirely mathematically. And it was much later when a group of astronomers were able to prove that, okay, general relativity actually works. And I've shown you a couple of more, and I've shown you a couple of examples where biology, as it seems, can also be thought of as a model. You can consider a cell, or a protein, or a system of proteins, or simply a phenomenon of any cells or genes and the like to be a model. And if it is a model, can you use mathematics to explain a lot of other things? So, for example, can you go into a more applicative domain like cancer, or can you explain wound healing, or simple cell migration? And as it happens that you can very effectively use mathematical models to explain a wide range of such phenomenon. And as it also happens that while doing so, you will realize that one overarching phenomenon is manifesting itself in so many different areas. Right. So this example is basically I'm going to talk about a project which a few of my professors here in IISc are involved in, and I'm very fortunate to have an exposure of that. So I'm gonna discuss about epithelial to mesenchymal transitions in cancer. So as it happens, and I'll give a brief primer on how cancer and metastases work. So as it happens is that a tumor, it grows in a particular site, and then through the bloodstream, it sort of flows around all over the body, and most of the times you end up having a case which is known as metastasis. Now, while these cells are sort of traversing from a primary location, from the primary tumor to different regions, they can do so in one or two ways. They can either leave as single cells, or they can leave as clusters of cells. And why it is important to distinguish between these two is because clusters, although you'd find them sort of going around and leaving the primary side very rarely, but even if they do, they are a higher chance of metastasizing. Is anyone saying something? Did I miss out something? Maybe not. I'll just check once. Okay, cool. So as I was saying that the clusters, the cells that live as clusters have a high propensity to metastasize more. And that begs the question is that what is the mechanism? Because if you know the mechanism of something, the next step would be to design solutions to treat it. So understanding things from a mechanistic point does become very, very crucial. Now, this is going to be where I'm going to cite a study which is done by one of our professors. So, cells were observed both in a primary tumor and the ones that metastasize, and it was found that during this process, when cells are leaving their site and going about through the bloodstream, they undergo a transition known as an epithelial to a mesenchymal transition, where cells which are usually epithelial they end up being mesenchymal. But that is for cells that leave. That is for cells that leave as single cells. For the ones that are clustered, you've got a very weird mix where these cells are neither epithelial or mesenchymal, but a mix of two. That's why they can stick around, thus having the epithelial property, but at the same time still go about the bloodstream, thus having a mesenchymal property. Similar cells are also found in wound healing. So the cells that are responsible in wound healing, they also have a very sort of a peculiar state where they are a bit of both. So maybe if you can understand the mechanism of the EMT in cancer, that would be worthwhile. That would make it worthwhile to look forward the same process in wound healing. So now I'd like you to observe the diagram or the network on the top. So on the network, you've got two proteins, and then you've got two miRNAs, microRNAs. So I won't go into the details. Just to let you know that the arrows with the pointed heads mean activation. The arrows with the blunt heads mean inhibition. Although it doesn't mean a lot in our discussion so far, but it's just a general trivia. So one key point from this network is that when the snail and the zeb, as you can see, when they are high, you will have a lot of mesenchymal cells being produced. However, when they are low, you'll have a lot of epithelial cells being produced. And you can design a lot of differential equations, like I had mentioned before. You can do all of that. You can do a lot of calculations. And once you're done with them, you will get, and if you do some graphs and you could draw some curves, you will get something like the image on the bottom half. So the image on the bottom half is basically trying to show the effect of the two proteins. Okay, sorry, not the proteins, the snail and the zeb mRNA on the state of the cell. So if you look on the extreme left, if you have a lot of snail protein, you could see that there is a single blue line. That is, if you look at the region from 0 to 180 on the x-axis, that is the snail proteins are very less, you'd find a lot of epithelial cells, and you will only find epithelial cells. However, if you increase it beyond 220, around 230 or so, again you will having a single solid line, and those cells are the mesenchymal cells. However, if you have somewhere in between, you can see that there are three lines in some cases. For example, if you look at 200, you are having three solid lines, one on top of the other. And that shows that in this region of the snail, you have a probability of observing cells which can be showing properties of both epithelial as well as mesenchymal. You can observe the same thing if you observe the y-axis. So when there is very less zeb, you can find again it's only epithelial. However, when it's too high, you can find it's mesenchymal. And in a very stringent region of both the proteins, I'm sorry, both the protein snail and the zeb mRNA, you're going to observe this weird case. Okay, maybe not weird, but this rather interesting case where you have cells that have properties of both uh, epithelial and mesenchymal. And as you would see is that these cells are going to be very robust. That is to say that if you can mathematically show using equations that these cells are going to be very robust using something known as stability analysis. Now, I'm not going to go into stability analysis, that's a big portion of differential equations. But what stability analysis shows you is that you've got two types of points. You've got equilibrium points and non-equilibrium points. Equilibrium points are those where if you perturb a system, it would still sort of come back to where it came from. And non-equilibrium points are those where if you even give a slight disturbance, it would sort of get out from that state where it was. And it was experimentally found that so these cells are basically the ones which are somewhere in between, the ones that from the cluster. And it was found that after even multiple passages of these cells, they are still the way they are. So they are very robust. So this proves that the mathematical treatment that was done to explain the effect of the molecules and the proteins on on the outcome of the cells indeed work, because you not only have a theory but you also have ways to experimentally test it out. So this is most this is mostly sort of a brief overview on how mathematics is sort of pertinent. I've tried to limit myself to very basic mathematical theories and very basic cases, which I hope would be easy to understand. There are also multiple cases I can talk about where you have to use data science and image analysis, even physics. I mean, I haven't discussed a lot of them because of the interest of time. So this is mostly about the math that, or the mathematical portion that goes beyond it. I'd also like to share my experience, or as to what qualifies me to even talk about this, is that I'm currently a part of a group, and I'm doing my master's thesis on a phenomenon known as immunological synapse. Now, I'm pretty sure if you are a biology student at any point of time in your life, you have heard of T cells and antigen presenting cells, and you know that T cells need activation, and for that they would need some other cell like dendritic cells, and there could be a few other cells I'm not able to recall at the moment, but those cells need to sort of come closer to the T cell and activate it. Now, you cannot do it without the T cell and the dendritic cell sort of coming together and forming a synaptic structure and staying there for a while. But, and to add on to the problem, is that T cells are not very static, they're highly dynamic cells. So they always keep on moving. So what happens is that the actin cytoskeleton in our body, in our cells, to be more precise, they organize themselves into two forms. If you can observe on the top right, you've got something known as foci. So you can see those really dot-like structures on the extreme right end of the picture there. Those are white, more bright spots out there. So those are the foci, and they act sort of like nails that sort of pin the cell down into a very stringent condition. But there are also another form of effectin that are branched actin, and they form something called lamellopodia. Now, these lamellopodia try to exert the opposite effect, where they try to sort of take the cell away from a static position. So there's a continuous competition going on between the foci and the branched actin. And depending on which concentration you have more, either of the one wins. What I'm working on at the moment is to see that since a cell is a limited system, that is, if you observe a cell in vivo, the cell doesn't have a continuous supply of its resources. It's a resource limited system, to be more specific. So, how is this resource limited system sort of distributing itself into these two forms? And what are the mechanisms and the equations that are governing it? And if so, can we sort of take use of that, and maybe we can explain a lot of conditions when you don't have an immunological synapse, or when you're having an immunological synapse that is sort of prolonging its lifetime. So can you explain those cases as well, using simply the fact that a cell is resource limited? So that is how I'm sort of associated in this domain. I'm doing my project work in this field, and I'm also very new in this field as well. Like I mentioned, that from my 4 years of my bachelor's, which I did in biotech, I hardly had any idea that you can actually uh, perform research, or you can even think of questions like these. So it's only in the past 1 year and 1.5 years when uh, really I've been able to get a full view of, or even be aware of, that such an area even exists. I think for people who are from maths or physics background, it is a bit more accessible for them. But for people from biology background, I think it's, at least for based on my experience, I feel not a lot of people get to know a bit about it. So I thought maybe it would be worthwhile to sort of bring this area to light, and hopefully this would, if it could, I'm really glad, motivate you to sort of think about this area, think about this field. Even if you are from a very biological background, you can sort of learn a lot of math, and maybe use it in your research if you have a research problem in your mind, or be part of a group. And the more importantly is that this isn't very theoretical, like you observed in the case of the cancer thing, is that you can design a theory, and at the same time you can test it out in a biological setup, using experiments which won't be very complicated. So that is mostly what I intended on sharing. These are a couple of videos which I would like you to go through if you can. I've mostly sort of, these videos are really helpful, would help you get a good idea, a taste in this field even better, because these are actually made by professors, not like novice students like me. And well, I suppose that's mostly from my end. I really appreciate the fact that you guys actually stayed and had the patience to bear with me. So really, thank you for your time. And if you have anything to ask, or if you want to share something, I'd be really eager to hear from your end. So that's mostly from my end.
Thank you, Shounak. If anybody has any questions at all, we're open to questions. Akshay, are you there?
Yeah,
Okay, you can take over.
yeah. I request all the participants to just switch on their videos so that we can take a snap. It would be well and good if you can switch on your videos.
Can all of you just switch on your videos for a second so we can just document it just for a second? It will be nice, all of you. All right. Thank you. Actually, that's it from my side. You can carry on.
Thank you everyone for joining us today and making the session such a successful one. It's just a reminder to all the participants that you'll all be receiving your certificates in your mail, so please have an eye on that. Now, coming to my responsibility, a heartfelt thanks to our speaker, Mr. Shounak Sanyal, for such an enriching and insightful session. Your journey, your achievements, and the knowledge you shared today have been nothing short of inspiring. One thing that truly resonated with me about you is your passion and knowledge in computational biology and your interest in sharing this knowledge with us. So, it's this kind of wisdom that makes session like these so valuable. So, on behalf of everyone here, let me take this opportunity to thank you for sharing your knowledge and your generosity in doing that. We are truly grateful for that. And coming to the chief guest, Dr. Deepthi and Dr. Lini, thank you so much, ma'am, for joining with us for the webinar. Once again, thank you to all the participants for your active involvement. Have a wonderful day ahead, and we are hoping to meet you all like this in future events. Thank you.
That's the end of the meeting. Yeah, thank you so much, Akshay, for hosting. It's been
Thank you.
wonderful. If you all have any feedback to share, you may share it now, or the meeting is over for you. Thank you all for joining again. Thank you so much, Shounak. Thank you so much for speaking today.
Yeah, thank you for